Hello everyone, welcome back to Sowkya Vision. Today we are going to start with the new topic, blood. The first chapter, plasma proteins. Yes, what are the plasma proteins? These are the proteins that are present in the plasma. So what is plasma? Plasma, it is a clear straw colored fluid portion of blood. Plasma is present in the blood. Yes, the proteins which are present in plasma are known as plasma proteins. Yes, now here the total volume of the blood in human body is about 4.5 to 5 liters. Yes, the total blood volume in the body is 4.5 to 5 liters. Now, the plasma from the blood is separated as what? Supernatant. So, plasma is separated out as supernatant blood cells remaining in the bottom of the centrifugation. So the plasma is separated from the blood and blood cells are also separated by the process of centrifugation. Yes, the plasma is separated as supernatant, whereas blood cells, remaining blood cells are settled bottom. Yes, now what is the composition of plasma in blood? So it is the 55 to 60% of blood. So blood containing 55 to 60% of plasma. Yes, remaining is blood cells. And now let's see the introduction of plasma. As I said, plasma is a clear straw colored fluid. It is a clear straw colored fluid. Yes, it is present about 55% of total body volume. Yes, there is 4.5 to 5 liters of blood in the human body. Of that, 55% is plasma. Yes, now the plasma which is defibrinated is known as serum. Yes, the plasma which is defibrinated, the defibrinated plasma is known as serum. When we call the plasma as serum, after the removing fibrinogen and prothrombin. After removing fibrinogen and prothrombin, we call it as a serum. Yes, so serum is plasma minus clotting factors. Yes, these two are the clotting factors. Yes, fibrinogen and prothrombin, which are involved in the formation of clotting. Fibrinogen is converted into fibrin by thrombin, which is activative form of prothrombin. Yes, after removing fibrinogen and prothrombin from the plasma, it is known as serum. Yes, so the components present in the serum as known as serum albumin or serum globulin after removing fibrinogen and prothrombin, which are the clotting factors. And now let's see the composition of the plasma. Now, plasma containing water and the solids. Majority is water, 91% is water, and the solids are 9%, of which 1% is inorganic molecules. Yes, and 8% organic molecules. Yes, now the plasma proteins is 7 to 8% of plasma. See, the plasma containing plasma proteins of 7 to 8%. 7 to 8%. Yes, now what is the concentration? Let's see, 6.4 to 8.3 grams per deciliter. Remember this value. Yes, 6.4 to 8.3 grams per deciliter. Now what are the plasma proteins? Yes, there are three main plasma proteins: albumin, globulin and fibrinogen. Yes, albumin is about 3.5 to 5 grams per deciliter. Remember the unit, you have to mention the unit along with. And globulin is about 2.3 to 3.5 grams per deciliter. Yes, and fibrinogen 200 to 400 milligrams per deciliter, which is very less compared to other two. Albumin is more. Yes, and now what is the albumin and globulin ratio? Yes, it ranges from 1.2 is to 1 to 1.5 is to 1. Yes, now where these are synthesized, where plasma proteins are synthesized. See, almost all plasma proteins are synthesized in liver except immunoglobulin. Immunoglobulin are the type of globulin, yes, which are involved in immunity. Yes, so almost all plasma proteins are synthesized in the liver. Now, how plasma proteins are separated from each other? How they are analyzed? Let's see with different process. See, there are different processes for the separation of plasma proteins. Yes, see, precipitation by salting out method. Precipitation by salting out method, Cohn's fractional precipitation method, electrophoresis separation of protein fractions, immunoelectrophoresis, ultracentrifugation technique. See, these are the main process used for the separation of plasma proteins. Yes, now let's see each process in detail. See, precipitation by salting out method. Yes, here we use a salt. Yes, here we use salt. See, first of all, we have to take the plasma. We have to prepare the plasma by removing fibrinogen. So, see, the function of fibrinogen is what? It helps in the coagulation. It helps in the clotting. Yes, it helps in the formation of clotting. So, we have to remove the fibrinogen priorly. So, after removing the fibrinogen, we need to add a salt. Yes, we got a plasma. We have removed the fibrinogen. Now we have to add the salt. So, the basically commonly we used salt is ammonium sulfate. Yes, the commonly used salt in the precipitation by salt out method is ammonium sulfate. And now the plasma proteins are aggregated. After addition of ammonium sulfate, the plasma proteins are aggregated and precipitated. Yes, now they are allowed for centrifugation to separate precipitated proteins from supernatant. Yes, the plasma proteins are supernatant. They are separated from supernatant. Yes, the proteins are again recovered for the further analysis. Yes, and now next process is Cohn's fractional precipitation method. Here ethanol is selected at different concentration. Take 3 to 4 samples at different concentration and doing fractionalized plasma proteins. Yes. And now the third process is electrophoresis separation of protein fractions, in which plasma proteins are separated. Plasma proteins are separated into different fractions by different electric charges. Yes, and now immunoelectrophoresis technique. Yes, it uses antigenic and electric charge. Here the plasma proteins are separated from each other by antigenicity and electric charge. And the last process is ultracentrifugation technique, in which the plasma proteins separated in buffered solution. Yes, then separated by what? Sedimentation. Separated by sedimentation. By different properties. By taking different properties like weight, shape and density. Yes, see, these are the different process for the separation of plasma proteins. Yes, precipitation by salting out method, Cohn's fractional precipitation method, electrophoresis, immunoelectrophoresis, ultracentrifugation technique. You have to study this. Yes, and see, let's see the synthesis of plasma proteins. See, in embryo, the plasma proteins are synthesized in mesenchymal cells. Yes, in the adult, these are synthesized in reticuloendothelial cells of liver, spleen, bone marrow. So, what is the first plasma protein that is going to be synthesized? That is albumin. Albumin is the first plasma protein that is going to formed in the human body. And now, what are the functions of plasma proteins? Now, coagulation of the blood. Yes, the plasma. What are the plasma proteins? Albumin, globulin and the fibrinogen, and fourth one is prothrombin. The fibrinogen's main function is to coagulate the blood, to clot the blood. The fibrinogen is converted into fibrin in the presence of thrombin. So thereafter forming a fibrin thread. Yes, this process is known as coagulation. We will study the coagulation of blood in the further classes. So here the first prior function of the plasma proteins is the coagulation of the blood by fibrinogen. Now, it is also have role in defense mechanism. Yes, it also involved in the defense mechanism. See, these are the gamma globulins involved in defense mechanism. Gamma globulins are the types of globulins. Yes, they are involved in the defense mechanism as act as antibodies. Yes, these gamma globulins acts as antibodies as they are involved in the immunity. They are called immunoglobulins. See, they are involved in defense mechanism of the body by what? Gamma globulins. As these gamma globulins act as antibodies involving in immunity, so they are called as immunoglobulins. And the third function is to transport mechanism, to transport the enzymes, hormones, different substances in the blood. So the main proteins that are carrying substances are albumin, alpha globulin and beta globulin. Yes, these two are the type of globulins. So the metals are carried by alpha and beta globulins. Yes. Now the regulation in acid base balance. The main plasma protein that is involved in acid base balance is albumin. Yes, albumin is the main plasma protein that is involved in maintenance of acid base balance. Now, as these proteins have buffering action, yes, the plasma proteins have buffering action. The buffering action of these plasma protein, especially albumin in the blood, is about 15%. Yes, the buffering capacity of the albumin is about 15% of blood. Yes, now let's see the another function, viscosity in the blood. So the plasma proteins provide viscosity. Yes, which provides viscosity, which is important for what? Maintaining blood pressure. Yes, the viscosity is important for the maintenance of blood pressure. And the maximum viscosity is provided by albumin. The maximum viscosity is provided by albumin. The maximum the acid base balance is also regulated especially by the albumin. Yes, now let's see some more functions of the plasma proteins. Next one is maintenance of oncotic pressure. Yes, as the plasma proteins are large molecules. See, plasma proteins, in general proteins are the large molecules which cannot pass through the capillaries. See, these are the capillaries. Let's see. Very small capillaries. The plasma protein size is this much. So these protein cannot enter to the capillaries. Yes, as they cannot pass through the capillaries at the tissue level. Yes, they exert oncotic pressure. Yes, the plasma proteins exerts oncotic pressure. So the net filtration. Yes, the net filtration is directly proportional to hydrostatic pressure minus oncotic pressure. Yes, so here also the albumin, especially the albumin, exerts 70% of oncotic pressure. Yes, now let's see the another function, erythrocyte sedimentation rate. Yes, the plasma proteins are also involved in erythrocyte sedimentation rate. The globulin and fibrinogen helps in the Rolex formation, which is the property of red blood cells. Yes, the Rolex formation is by globulin and fibrinogen, which is involved in the formation of ESR. Yes, which is helpful in diagnosing and different tests and different examinations and tests. And another function is suspension stability. What is suspension stability? See, if this is the blood vessel, I think you can see this. Yes, this is the blood vessel. The RBC are equally distributed, equally suspended by plasma protein. This process is suspension stability. Yes, so these RBC are equally uniformly suspended by globulin and fibrinogen. Yes, globulin and fibrinogen are involved in suspension stability. And it also involved in production of tryptophan. Yes, tryptophan, it is a substance. It is a substance produced by leukocytes. Yes, which gives nourishment to the cells in culture. Yes, it gives nourishment to the tissue cells in culture. And the last function is reserve protein. See, when you are doing fasting or you don't have adequate amount of protein food, so these plasma proteins used as source of energy. Yes, so these plasma proteins are splitted into amino acid by macrophages. These macrophages converts the plasma proteins into amino acid which enter the blood and circulated throughout the body to give energy. Yes, these are the functions of the plasma proteins. Let's see the applied. What is hyperproteinemia? If there is increase in protein concentration, plasma protein concentration above the physiological level, it is known as hyperproteinemia. If there is any decrease, it is known as hypoproteinemia. When it will be hyperproteinemia? During the conditions of dehydration and hemolysis, there will be hyperproteinemia. And during diarrhea and hemorrhage, there will be hypoproteinemia. Yes, this is the complete discussion of plasma proteins. If you have any doubt, please do comment in the comment section. If you like the video, please do like and please do subscribe and support the channel. Thank you.